Can one of you tell me if you have already had maybe in the last semester some online examinations and the mode of conduct, which sites were used or what was the platform? I'm trying out some platforms for your quiz. So if you can help me or suggest some, yeah, Vishantan, tell me.
Ma'am, the quizzes were most probably taken on Moodle. The subjective tests were via Gradescope.
Subjective or via? Sorry, Vishantan, you're cracking.
Ma'am, subjective were via Gradescope. Gradescope.
We score and objective or by
Moodle.
Moodle. Okay, because I've never used Moodle. I know about exam.net. And I thought maybe even I could use Google, but I'm just certain sites. Anyway, ठीक है हम देखते हैं और भी कुछ सजेशन हो तो मुझे बता देना तुम लोग. Let's first start with what we did in the last class, and I would like to first ask you if you have some queries. Otherwise, I'm going to ask you. Last class से किसी को कोई question, कोई confusion? Basically, अभी हम लोग सिर्फ cell structures की बात कर रहे हैं, तो इसमें सिर्फ I'm just describing to you what are the different structures, and this is exactly what I'm going to do today as well. Though it will be a little faster today because most of you would be aware of the eukaryotic cells and the different organelles. तो चलो कोई confusion नहीं है तो हम पूछते हैं कि about gram staining that we did in the last class. Can one of you tell me what would happen if we let's say miss out the decolorization step? What would be the result? Anybody? विशंतन पहले मुझे देखने दो कि बाकी लोग जगह हैं कि नहीं जगह हैं. अंकित राज अंशु, you were one of the first ones to join. बताना जरा.
Yes, ma'am.
Tell me.
Hello.
Tell
Am I
me
audible?
what would happen. Tell me, Ankit. Yes, you're very much audible. Tell me.
We cannot distinguish.
Why? I'm asking you why. Jatavat, Pavan, Nayak. Ashi, hold on, I'll ask you. Let me check how many of them are in front of the laptops. Jatavat, Pavan, Nayak, please unmute yourself.
Hello,
Okay.
ma'am. Hello, ma'am. Hello, ma'am.
Hello?
Hello, ma'am.
Yes.
Ma'am. Hello. I'm audible, ma'am.
Pavan, tell me. तो फिर क्यों म्यूट कर लिया?
Oh, ma'am. It's by mistake. It's by mistake, ma'am. Ma'am, when we add color to it, they are of same color. Like they are two are of same color, purple and purple. So we need to like the gram positive cell. When it is decolorized, it doesn't get decolorized due to the wall present in the gram positive cell. So we try to decolorize it so that the
So if you
negative
don't add the decolorizer, if you don't add, what will happen? The decolorizer.
Ma'am, can you repeat again? Hello. Hello, ma'am. Hello.
Tell me.
Yes, ma'am. What did you say? Yes, ma'am. What did you say?
I'm sorry, I could not get you. Please tell me what will happen if you don't add the decolorizer.
Ma'am, if we do not decolorize it, we can't find difference, ma'am. Since they are of both purple color, we need to decolorize so that one becomes white, and when we add iodine, so it becomes a lightish pink color, so that we can differentiate the both things.
Okay. Ashi and Vishantan, you want to add something?
Yes, ma'am, it's not actually iodine. We add saffron in so that it becomes a light pink color. And like after decolorization, the gram-negative bacteria become like colorless. So afterwards, if we add saffronin, since the colorless one doesn't have a purple dye, so it shows a light pink color.
Yeah. आशी कुछ ऐड करना है?
Also, ma'am, the lipid layer will remain intact in this case because we're not adding alcohol or alcohol and acetate.
अच्छा, what happens आशी if we don't add the mordant? तब क्या होगा?
Ma'am, the mordant to make sure that uska color aa jaye, like it shows the color. Iodine is added for this purpose.
नहीं नहीं, कलर तो वो है ही ना. फर्स्ट जब तुमने क्रिस्टल वायलेट डाला तो पर्पल कलर हो गया. फिर मॉर्डेंट अगर हम मिस करते हैं, कशिश,
So
can
that
you
it
tell me what is
मैम इट मेक श्योर मतलब
आकाश-
पीजी वाली लेयर में इट मेक श्योर द मॉडर्न मेक श्योर कि वो कलर सीप कर जाए अंदर इन द केस ऑफ ग्राम पॉजिटिव.
Okay. Anybody who wants to add something, modify her answer, whatever. I'm asking what will happen if you do not add the mordant? If you miss the second step, what is going to happen? नरेंद्र कुमार बताएंगे. नरेंद्र अनम्यूट करना अपने आप को.
Hello, ma'am.
Yes, Naren, tell me.
Ma'am, actually, I don't know what happens if we don't add the modern. But ma'am, the last question you asked, I know about that.
Tell me
Ma'am,
about that then.
ma'am, in decolorizing that the thick cell wall that dehydrate and shrink because of decolorization.
Correct, that is correct. हाँ, बाकी सबने ग्राम नेगेटिव का बताया, तुमने ग्राम पॉजिटिव का बताया कि it will shrink further and it will help retain the color. I mean, it will not move out. अब सोचो कि क्यों मॉर्डेंट अगर नहीं ऐड करेंगे तो क्या होगा? आयोडीन का क्या पर्पस है? Vishantan knows it, Justine knows it. Hold on. I'm asking Narendra. बोलो. चलो ठीक है, बाकी लोग से पूछते हैं. Okay, Justine, tell me.
Modern binds the dye so that it may not be removed easily.
Vishantan?
Ma'am, I guess it basically forms a complex with the crystal violet that we added in the first step. And again, that increases the binding, so decolorization where it doesn't like get removed.
So, the crystal violet has been able to diffuse through the cell wall of both gram positives and gram negatives, and it will diffuse back if you do not add something to capture it inside the cell or help it retain. So, iodine is only making larger molecules, I mean complexing with crystal violet so that it's not able to move out of the cell. It stays in the inside the cell. Okay. और कुछ कोई और क्वेश्चन है तो हम फिर आज की क्लास शुरू करें. ठीक है, I don't see any hands raised. Okay. So today we will talk about the structures found in a typical eukaryotic cell and their respective functions. Now you've studied the basic organelles during your school time, so this will be like a recap. We'll move a little faster and a little in depth as well. But please ask me questions whenever you think there's something which is not clear. उसी समय पूछो मुझे. Last के लिए wait मत करो, हाँ. Now, before we talk about each organelle, here's a quick recap of some of the points that we talked about when we were discussing the prokaryotic cell structure. There's certain common features between the two, like the genetics, the plasma membrane, the cytoplasm. You saw that both have ribosomes. Then some of the appendages like flagella are also common in prokaryotes and eukaryotes. Cell division, while it is mitosis and meiosis in eukaryote, it is binary fission in prokaryotes. This we will also discuss when we talk about bacterial growth. We saw how the ribosomes were different in the two. So one was 70S and the other one was 80S, 80S being the bigger ones. We will start with the nucleus in the eukaryotes. The genetic material in eukaryotes is compartmentalized in a double membraned organelle called the nucleus. Now, both these membranes are phospholipid layers, the unit membrane that we talked about earlier. There is a perinuclear space between the two membranes. Now, whenever I say there is space, you have to remember that these are not empty spaces. These are actually sites of action for various steps. The two membranes of the nuclear envelope, they, as you can see here, they fuse at sites to form nuclear pores. And these pores, they control the passage of several ions and even RNA across the outer membrane is in continuation with the endoplasmic reticulum. If you remember, I mentioned that in eukaryotes, the transcription and translation, that is the central dogma, are spatially and temporally separated, unlike in prokaryotes. In fact, prokaryotes can't afford to have a separation as there is little compartmentalization. Transcript in the eukaryotes, the mature mRNA travels outside the nucleus and it is in the cytoplasm that the next step, that is translation, takes place. The matrix inside the nucleus is called nucleoplasm. This is where the chromatin lies. Now, abhi tak humne chromosome kaha tha. Chromatin is the relaxed form of a chromosome, while when DNA is tightly packed and organized, it is called as chromosome, which are of course much condensed forms. Now, besides the genetic material, there are distinct structures called as nucleolus, which are primarily made of RNA and proteins. This is where ribosomes are synthesized. And a nucleolus, you will see it is visible when the cell is not dividing. Some nuclei are known to have even more than one nucleolus. Please remember that there is no membrane enveloping the nucleolus. Then the nuclear pores, they function as selective gates. In fact, they are massive proteinaceous complexes. They also have a role to play in maintaining the integrity of the nucleus by preventing the free flow of macromolecules across this nuclear membrane. The NPC or the nuclear pore complex has been observed to have 8-fold symmetry. Also, it has rings located on both the cytoplasmic side as well as the nuclear side, nucleoplasmic side. It contains a structure similar to a basket which extends into the nucleoplasm. This is called as the nuclear basket. And there are filaments protruding out into the cytoplasm called as the cytosolic fibrils. Now, towards the inner, inside of the inner membrane, you will see that there are some cytoskeletal elements forming what is called the nuclear lamina. Now, the proteins that form the lamina, they are called lamins, and this is a fibrous mesh for structural support to the nucleus, the membranes. With respect to the genetic material, in eukaryotes, the blueprint, that is DNA, is concentrated in the nucleus as linear chromosome, unlike the circular chromosome that we saw in prokaryotes. While in prokaryotes, there was only one chromosome, in eukaryotes, there is diploidy, which means 2 copies of each chromosome. That is a diploid number. While gametes, so I don't know, tell me how many of you know the number of chromosomes in a human cell, please? How many chromosomes are there? Narendra, batao.
Ma'am, I think 46.
I think नहीं. तो 46 क्या है? 46 different chromosomes है?
Yes, ma'am.
एक गैमेट में कितना होगा? एक ह्यूमन गैमीट में कितना होगा?
23 पेयर्स।
ठीक है, तो 23। हाँ विशंतन, कुछ बोलना है, बताओ।
No, ma'am.
हाँ, so the N is 23 in humans, and because we are diploid, you will see that a human cell has 46 chromosomes. ठीक है, that is N number. The one set is N, and in prokaryotes you have only one set of genes, so one chromosome. Okay, the DNA is tightly wound in an organized manner on proteins called histones. Approximately 200 DNA base pairs. They wound around 8 histone proteins and make up the smallest unit of DNA packing called as the nucleosome. So these are various levels in which it is compacted. Okay, the nucleosome, as you can see the structure, they appear to be like beads on a string. So in fact, this model is called as the bead on a string model. This structure further forms supercoils, and further loops are formed to condense it tightly. This is the kind of structure that you observe for the figure on the towards the end that you observe for human chromosomes as well. You will study the genomic organization in detail in your molecular biology course in the next semester. I've again included the same comparative table to differentiate between pro and eukaryotic chromosomes for ease, because probably you'll be able to appreciate it here better. Now, eukaryotes, they have an extensive endomembrane system, which is a collection of membranes and of organelles that work together to synthesize, to modify, package, and then deliver various macromolecules like proteins, carbohydrates, and lipids to different organelles or sites where they are required. There is an efficient pathway or a sequential order in which the synthesis and modifications occur. This is termed as the biosynthetic secretory pathway or the biosynthetic secretory route. I will cover the organelles as in this sequence of traffic. Now, you saw in the last slide, the outer membrane of the nuclear envelope is in continuation with flattened sac-like structures called the endoplasmic reticulum. As the name suggests, it's a network found within the cytoplasm. Depending on the type of ER, lipids and proteins are synthesized, and then through transport vesicles, they are transferred to the next organelle, which is Golgi complex, where they are modified, appropriately tagged, packaged, and then sent off to their destination. So this is called the exocytic pathway, wherein cargo moves in the sequence in a eukaryotic cell and ultimately moves to the target or the destination. Besides this, there's also a pathway to bring in nutrients from the outside. Now, starting with the endoplasmic reticulum, that is right after the nucleus in this pathway, it is considered as the largest organelle in the cell. It's an interconnected network of tubules and flattened sacs throughout the cytoplasm in a eukaryote. It is a major site for protein synthesis and transport, then protein folding, lipids and steroid synthesis. It is also involved in metabolism of carbohydrates, and another important function is its storage of calcium. Now, depending on whether or not ribosomes are attached to these flattened sacs, also called as cisternae, they are classified into either the rough endoplasmic reticulum or the smooth endoplasmic reticulum. The former, as you can see, because of the impregnation with ribosomes, they have a rough appearance. Now, while some proteins are made by free ribosomes in the cytoplasm, those that are destined for secretion, they are made on ribosomes which are bound to the RER. These are the proteins which undergo a series of post-translational modifications, including tagging to be directed to the correct destination. Now, this interaction between the ribosomes and the RER, this is dynamic. It goes through a period of attachment as well as detachment depending on the cell needs. In fact, one can judge the status of the cell by the type of ER present extensively. So a cell that will produce a lot of secretory proteins will have a more extensive RER compared to the smooth endoplasmic reticulum is important in the synthesis of lipid kinds, phospholipids, steroids, etc. Just tabulated what I just said, that is the differences between SER and RER. From the endoplasmic reticulum, there are membrane enclosed vesicles which pinch off to shuttle the cargo to the next organelle in the pathway, that is the Golgi apparatus. The Golgi complex is again a membrane enclosed organelle. It is a series or a stack of such flattened sacs, and as we called it in the endoplasmic reticulum, they are called as systems. The stack has a distinct polarity in terms of both its structure as well as function. So there are two phases. You will see there's a cis phase and there's a trans phase. Cis phase is the entry phase which receives the cargo from the endoplasmic reticulum, and the trans phase is what faces the plasma membrane, where the finally modified and packaged products, they exit to reach the destination. The intermediate Golgi. Now, as the content moves through the Golgi stack, they are processed and sorted. You can imagine this as a postal network, basically sorting out things, tagging, and then taking it to the destination. The movement here occurs through pinching off in the form of transport vesicles and then fusion to the next cisternae. Now, what actually happens in the cisternae? Each cisternae contains a different set of enzymes. And when I say modification, I am referring to, for example, addition or removal of sugars from proteins called as glycosylation. There could even be modifications, for example, addition of sulfate groups or phosphate groups. Now, these modifications, they occur sequentially as the cargo moves from one cisternae to the other. There could be other kind of modifications, for example, adding of signal molecules to the cargo to direct it to the destination. There could be, for example, some proteins which are deposited in the plasma membrane. Others would be for any other organelle, let's say. So the entire endomembrane system involved in the biosynthetic secretory pathway is a very dynamic system with continuous pinching off and fusion of vesicles. And finally, from the Golgi complex, secretory vesicles carry the cargo to the destination, like you see here, fusing to the next membrane. Now, external, we have talked about the plasma membrane, that it has a unit structure. We briefly covered. Kashish, tell me.
Ma'am, how does they reach the site? Means how they do identify that where they have to reach, or how to move? Basically.
How they move, matlab?
Means like the
What, the
undergraduate.
cargo?
Yes, cargo.
Acha, okay. So there are two models. I did not want to mention it in that depth. However, now that you've mentioned, there is one model is the vesicular transport, and the other model is a cisternal maturation model. Now, the vesicular transport is what you saw in the last slide. Just make after a modification, these are pinched off. Once that enzymatic reaction takes place, let's say glycosylation ho chuka ya phosphorylation ho chuka, in this sequential manner, it is pinched off from the ends and it fuses to the next cisternae, jahan uska further modification hoga, which could be any of these additions, koi modification is tarah ka, ya fir it could even be tagging, कि हमने एक signal sequence लगा दिया उसमें, जिससे कि we know कि this signal will only be taken to the mitochondria or it will only be taken to the chloroplast. ये तो एक मॉडल है वेसिकल से मूव करता है और गोल्गी के सिस फेस से ट्रांस तक आता है. लेकिन देयर इज वन मोर मॉडल व्हिच इज कॉल्ड एज सिस्टर्नल मैच्योरेशन. उसमें ये कहते हैं कि द एंटायर सिस्टर्ने मैच्योर्स by undergoing modification wherein the enzymatic makeup of that particular cistern changes. तो ये सिस्टर्ने जो यहाँ पे सिस फेस में है ये मैच्योर होके यहाँ और नया सिस्टर्ने फॉर्म होता जाएगा and it will ultimately mature with all the modifications and pinch off from vesicles and go to the desired destiny. ठीक है, तो दोनों मॉडल्स I would say they are kind of being debated कि कौन सा करेक्ट है. Anybody else? कशिश ये क्लियर हुआ?
Yes, ma'am.
एक और किसी का हैंड रेज था. विशांतन तुम्हारा क्या?
नो मैम, अभिनव.
मैम मेरा था अभिनव, यही क्वेश्चन था.
अच्छा अभिनव, हाँ बताओ. अच्छा यही क्वेश्चन था, ठीक है. समझ में आया ये दोनों मॉडल. हम इस तरह के बहुत सारे डिटेल में तभी जाएंगे जब तुम में से पूछोगे, ठीक है. वरना हम सिर्फ रिकैप कर रहे हैं जो आपने पढ़ा होगा एंड मे बी सम. ओके, अगर सॉरी अभिनव
मैम आपकी आवाज काफी कट रही है, काफी ज्यादा बीच बीच.
अरे पता है आज मेरा नेटवर्क बहुत खराब है. तुम लोग को जब हम साइट चेंज कर रहे हैं तो दिख रहा है ना?
यस मैम.
यस मैम.
चेंज दिख मोबाइल डेटा पे आ जाए क्या? I'll share it from my desktop and I'll is it that big a problem that I should move shift from the computer to the mobile data? Okay, तो जब ऐसी बुरी सिचुएशन होगी मुझे हैंड रेज करना तो मोबाइल डेटा पे आ जाएंगे, ठीक है. ओके, सो मूविंग फॉरवर्ड हम लोग एंडोसिटिक पाथवे देख रहे हैं जिसमें कि एंडोसिटिक पाथवे देख रहे हैं जिसमें वी सॉ कि न्यूक्लियोप्लाज्मिक रेटिकुलम पे चीजें आती हैं एंड एंडोप्लाज्मिक रेटिकुलम में वी सॉ दैट प्रोटीन सिंथेसिस होती है, लिपिड सिंथेसिस होती है. बहुत सारे प्रोटीन्स जो साइटोप्लाज्म में बनने हैं या बनते हैं उनको भी अगर मॉडिफिकेशन चाहिए तो वो एंडोप्लाज्मिक रेटिकुलम का रूट लेके एंडोप्लाज्मिक रेटिकुलम से गोल्गी कॉम्प्लेक्स पे आते हैं. वहाँ सारे मॉडिफिकेशन और टैगिंग होता है जिससे कि फिर वेसिकल्स अपने डेस्टिनेशन पे जाते हैं, ठीक है. ये वाला पाथवे क्लियर है. Coming to the plasma membrane, here the major difference is the presence of sterols in eukaryotes. जो हमने जिसकी बात की थी, sterols are also lipids, but they have a rigid ringed structure, and they have a role to play in maintaining the fluidity of membranes. Fluidity के बारे में हम लोग जब plasma membrane की बात करेंगे तो we'll take this up in detail. Now, external to the plasma membrane, there is a cell wall in some eukaryotes, only in some eukaryotes, okay, like protists and fungi. But the architecture is not as complex as that found in prokaryotes. Okay, it's a thick, rigid, and protective layer. The absence of the cell wall necessitates the presence of some means of support, especially for the organ. Okay. So eukaryotes, they have evolved a complex cytoskeleton consisting of different classes of molecules. Now, cell wall जब इसकी बात कर रहे हैं तो क्या मुझे कोई बताएगा कि what is the charge on bacterial cell surfaces and what contributes to the charge? बताओ. इसकी बात हम लोगों ने लास्ट क्लास में की हुई है. सेल वॉल से रिलेटेड.
Ma'am, is it negative?
It is negative. And क्यों?
Like I don't know. I just took a guess. Maybe because of phosphates.
हाँ, तो टेकोइक एसिड और एलपीएस याद है किसी को? आशी याद है? आशी?
Yes, ma'am.
टेकोइक एसिड कहाँ था? कहाँ प्रेजेंट था?
ग्राम पॉजिटिव के केसेस में.
और ग्राम नेगेटिव में
एलपीएस था,
है ना. और दोनों फॉस्फोरिलेटेड नेगेटिव दे रहा था चार्ज. हमने बोला था दे बोथ कन्फर नेगेटिव चार्ज. तो हमेशा बैक्टीरियल सेल्स जो है ना नेगेटिवली चार्ज होते हैं, ठीक है. द सर्फेसेस आर नेगेटिवली चार्ज. ओके, This was just to see कि तुम लोग में से कितने लोग अभी active हो. Okay. So cell wall is less complex in the case of eukaryotes, and cell wall is not a mandatory structure in the eukaryotes. And इसके लिए it is important कि जो cell wall के function थे, rigidity, integrity maintain करना, that is actually taken up by some of the other structures found in the eukaryotic cell. और उसमें pivotal है the cytoskeletal elements. The cytoplasm contains a range of fibrous proteins collectively called as the cytoskeleton. There are three types of fibers which make up the cytoskeleton. The microtubules, they are the thickest of the three with a diameter of approximately 20 nanometer. Microtubules, as the name suggests, are made up of the protein tubulin. हमेशा बायोलॉजी में तुम देखोगे कि नेमिंग जो होती है वो या तो स्ट्रक्चर या तो फंक्शन से रिलेटेड होती है. तो microtubules made up of tubulin. The other extreme or the finest ones are called the microfilaments. These are made up of the protein actin. So actin and tubulin make up microfilaments, microtubules and microfilaments respectively. As the name suggests, the intermediate filaments, they are of intermediate diameter, approximately 10 nanometer, and it is made up of a range of proteins. Now, the nuclear lamina that I mentioned earlier when talking of the structure of the nucleus, जब हम inner membrane के नीचे आपको एक layer दिखा रहे थे support के लिए, that is also a type of intermediate filament. The cytoskeleton gives the cell strength and rigidity, thereby helping to maintain cell shape. And एक और बहुत important जो role है cytoskeletal fibers का, that is to localize the organelles in the cytoplasm and thereby also offering strength. They also help in motility. We will see this in a while. Here is a comparative of the different kinds of cytoskeletal elements with some of their primary functions like motility and structural support. That brings me to the locomotory appendages in eukaryotic cell, which are made up of microtubules. Now, it's an intricate design compared to the more simplistic structure of flagella in prokaryotes. Eukaryotes में क्या होता है कि both flagella and cilia, they consist of 9 pairs of microtubules with side arms. These are the side arms in orange and blue that you see, which are made up of molecular motor dynein. Besides this, there is an inner pair of microtubule. Here you see them in the center. This arrangement is called as the 9+2 arrangement. And if you notice, the basal body which anchors the flagella does not have the central pair. And this arrangement is called as the 9+0 arrangement. The doublets, as you see the subfiber A and subfiber B, they are connected by the protein called nexi, and they are connected to the central doublet by radial spokes, giving it a structure of this sort. The complex of this microtubules is covered, whether it is cilia or a flagella, it's covered by a sheath which is in continuity with the plasma membrane. This is just another view to show you how things look like without the markings. This is a more simpler picture. The cilia, it is the structure, like I said, is quite similar to that of a flagella, but it is shorter and it is more in number in any cell. And also the type of motion that it exhibits is different in the case of cilia. Now coming to the cytoskeletal elements found in prokaryotes, I told you that I'll take this up when we talk about the eukaryotes. In some bacteria, there are elements found which are analogous to those that we talked of in the eukaryotic cell. So for example, this bacteria called Colobacter has 3 different kinds of cytoskeletal elements. Please remember that we told earlier, we discussed that a major difference between pro and eukaryotes is that prokaryotes lack a cytoskeletal element. Now here, there is a protein called FTSZ which performs functions exactly like tubulin and helps in cell division. So it constricts the cell in the center when the cell divides. Then there's another protein called MREB which functions like actin. So it has a dynamic localization. It is found across in the spiral mode throughout the cell, and as well as it would be found in the constriction when the cell is dividing. We will take these two cytoskeletal elements when we talk about the division of cell or bacterial growth. We will see how they facilitate division. But there's another prokaryotic protein called as crescentin, CRAS, which attributes the role is, in fact, the shape of the bacterial cell is attributed to the presence of this CRAS. So while there's a lot being done in identifying other cytoskeletal elements in prokaryotes and trying to understand the function of these cytoskeletal elements, in fact, Professor Preeti works on FTSZ and MREB in bacteria. There are still a lot of things which are yet to be understood here. So I just wanted to mention to you these three different kinds that we know of. Okay. Now coming to an organelle which you are well aware of, the powerhouse of the cell, that is the mitochondria. It is another organelle with double membrane separated by an intermembrane space. Now tell me कि अभी तक हम लोगों ने कितने organelles किए जिसमें double membrane देखा हम लोगों ने? Mitochondria में double membrane है. और कौन से organelle में double membrane होता है? अभी तक जो हमने cover किया है. हाँ आशी?
Nucleus.
और कोई?
I guess I remember nucleus ही.
ना एंडोप्लाज्मिक रेटिकुलम डबल मेम्ब्रेन है ना गोल्गी. अभिनव हाँ वो हम लोग आगे पढ़ेंगे, मतलब व्हाट आई कवर्ड टिल नाउ हम पूछ रहे थे. एनीवे सो शी वाज राइट, न्यूक्लियस होता है और इसलिए मैंने पूछा क्योंकि एंडोप्लाज्मिक रेटिकुलम या गोल्गी कॉम्प्लेक्स में सिंगल मेम्ब्रेन होता है. ठीक है. सो यहाँ पे भी आई मेंशन दैट देयर इज इंटरमेम्ब्रेन स्पेस. तो जहाँ पे भी हम स्पेस बता रहे हैं तो मैं समझ लेना है कि वो फंक्शनल एरिया है, नॉट जस्ट वेकेंट और लाइंग स्पेस, एम्प्टी स्पेस. The outer membrane is covered with porins. ये कहाँ हम लोगों ने सुना था? Porins. Porins, students. Students, पोरिन कहाँ था? लास्ट क्लास में तो किया था.
ग्राम नेगेटिव बैक्टीरिया
में आउटर लिपिड लेयर में. ठीक है. तो outer membrane of the mitochondria also has similar channels called as porins, while the inner membrane is drawn into these foldings or these bends, these invaginations called as cristae. अभी तक हम लोग एंडोप्लाज्मिक रेटिकुलम और गोल्गी कॉम्प्लेक्स में सिस्टर्ने कह रहे थे. यहाँ पे the folds are called as cristae. It is the outer membrane which is more permeable. The inner membrane has enzymes which synthesize ATP, जो आप पावर हाउस कहते हो कि एनर्जी करेंसी यहाँ प्रोड्यूस होती है. The interior of the mitochondria is called as matrix, and this is also a prime location for various reactions that take place. The unique feature of mitochondria and also of the next organelle that we're going to cover is the presence of genetic material in these organelles, which are circular DNA molecules. And you will find ribosomes in these organelles, which are of the 70S type. Another interesting point is that they divide independently of the cell by binary fission. Now I'm going to come back to all these points. But before that, I'm going to show you the similarity with the other organelle. The next organelle, which is the chloroplast, it is just one of the many types of plastids found in eukaryotic cells. Now, what are plastids? Plastids are involved in synthesizing and storing food. Chloroplasts are so called because they are chlorophyll containing plastids. They are responsible for photosynthesis. This is the next structure, like Abhinav said, this is a double membrane structure. But in addition to these two membranes, it has a very complex internal membrane system called as the thylakoid membrane. Now, the properties of the inner and outer membrane of the chloroplast is exactly similar to that of a mitochondria. So, the outer membrane would have porins like I told you for the mitochondria. The inner matrix here is called as stroma. The thylakoid membrane system is actually arranged in the form of these stacks or grana in the stroma. Each grana contains about 10 to 20 thylakoids. And the grana are connected to each other by stroma lamellae, this membranous system which is called as stroma lamellae. Similar to the mitochondria, the chloroplast has DNA molecules and also 70S ribosomes. What does this indicate, students? When I tell you that the outer membrane is like the gram-negative cell wall, the outer layer of the gram-negative cell wall, and I mentioned to you that there are circular DNA molecules in both mitochondria and chloroplast. To add to that, there is 70S ribosome. 70S ribosome कहाँ होता है? 70S ribosome किसका feature है? Is it typical eukaryote? हाँ भव्या?
मतलब वो पहले बैक्टीरिया थे।
मतलब वो पहले बैक्टीरिया थे, ओके। तो इसके पास अपनी मशीनरी है, अपना जेनेटिक मटेरियल, ठीक है, प्रोटीन सिंथेसिस के लिए इसके पास राइबोसोम्स है जो आपको नेसेसिटी था प्रोटीन्स बनाने के लिए। अपनी जेनेटिक इन्फॉर्मेशन भी है और सेल का स्ट्रक्चर डबल मेम्ब्रेन स्ट्रक्चर है, आउटर मेम्ब्रेन में पोरिन है। इन द नेक्स्ट क्लास आई वुड वांट यू टू टेल मी कि what these unique features in mitochondria and chloroplast indicate, not spontaneously. अभी तो तुमने कहा कि वो पहले बैक्टीरिया थे। I want you to read about it and the next class is going to be tomorrow. तो शुरू के 15-20 minutes you are going to tell me कि ये क्या indicate करता है with the mitochondria and the chloroplast having all these features similar to a prokaryotic cell. Please tell me what does this indicate. And you have to think about now, I mentioned this earlier also, we say that prokaryotes are devoid of compartmentalization and then we talked about all these structures that are present in the prokaryotes. We also say that prokaryotes मतलब cytoskeletal element absent. तो अब आप जब define करोगे prokaryot को तो कैसे define करोगे ये आप मुझे बताओगे next class में, ठीक है, including the unique features. You will study why or what are the grounds in, let's say, what is the significance of the mitochondria and the chloroplast having the DNA and having the ribosomes? Is it clear? अब तुम मुझे पूछो आज के टॉक में जो भी क्वेश्चन है या क्वेरीज है ये पूछो आप, because this is where we will start. We will, I think I will talk about, because I've completed prokaryotic and eukaryotic cell, I will now talk about the plasma membrane so that I can differentiate between the bacteria and the archaea, and then take up representative of a fungi, representative of a protist, so that we are then through with this unit. कोई डाउट नहीं है, सब कुछ क्लियर है? ऑर्गेनल्स तो जितना तुम लोगों ने स्कूल टाइम में कवर किया था उससे थोड़ा ज्यादा समझ में आया या बस उतना ही था या सब कुछ कवर्ड था तो तुम्हारा ये सब स्कूल टाइम में? कोई? दीपक जहांगीर अनम्यूट करें प्लीज। दीपक, ध्वनि, हाँ दीपक बताओ। दीपक ये सब स्कूल में कवर हो चुका था ना तुम्हारे? ये रिकैप था? क्या हुआ दीपक? ध्वनि, टेल मी।
Ma'am, school में we had covered, if we covered these topics, they were very basic and also it's been such a long time that we've covered these
तो
topics.
रिकैप हो गया।
Yeah.
तो अभी क्लियर है ना?
Yes, ma'am.
ठीक है, और कोई किसी को कोई क्वेश्चन? नहीं पता है, आई एम नॉट एबल टू पुट अ फेस टू योर नेम, सो मे बी जब नेक्स्ट टाइम तुम लोग आंसर करोगे या क्वेश्चन पूछोगे तो प्लीज अपना कैमरा ऑन करना, बिकॉज अदरवाइज तो हम तुम लोग को पहचान ही नहीं पाएंगे। प्रथम बंसल, इज एवरीथिंग क्लियर? ये तो अवे है।
Yes, ma'am.
हाँ प्रथम,
Yes, ma'am. Everything is
समझ में
clear.
आया?
Yes.
Okay, perfect. ठीक है, then we are going to meet tomorrow and we will start with a discussion wherein I told you I'll be asking you for points on these 3 questions. ठीक है, आप लोग मुझे बताओगे। ठीक है, then I am ending the class. Thank you all students.
Thank you, ma'am.